Welcome back to Chapter 2, Methods of Separating and Purifying Substances, Section 2D, Distillation. By the end of this video, you will be able to describe distillation, distinguish between simple distillation and fractional distillation, and recognize how to reduce risks when carrying out a distillation. If you have seen our video on pure substances, you will know that chemists use the word mixture in order to describe two or more substances not chemically combined together. For example, if we have 4 different colors of beads, in a mixture of these beads we can still identify each of the different colors and the essential qualities of each bead that is now in the mixture. They have remained unchanged, if you can see here, and the equivalent of the color of each of these beads that remained unchanged in this mixture is the chemical properties of each component, which are unchanged as we have said now. As a result, mixtures can be separated by physical means of separation, those that do not involve a chemical reaction. A really useful example of these for separating mixtures is distillation. We can start by thinking about simple distillation, which is used to separate out a liquid from a mixture. It is a common example to separate mixtures with different boiling points. A common example used in industry is to separate pure water out from seawater. The setup used to carry out simple distillation is known as the simple distillation column, as shown in this diagram, where all the key features have been labeled here. So you can see we have the distillation flask, or we can call it also round bottom flask because it has a round shape. Inside this round bottom flask or the distillation flask, we put the solution that we are aiming to separate. And up here we have this connection that connects this flask to the other parts of distillation. And up here we can insert the thermometer. This part is called the condenser. The condenser has 2 inputs. We have the cooling in part and we have the cooling out part. So here we allow water to enter to cool out the condenser, and from there the water will flow out. And we have here a flask that will collect our pure product. The mixture, which is in this case seawater, is placed again, as we said, in this distillation flask. And as a result, the component of this mixture with the lowest boiling point will evaporate first. So, simple distillation, again, it depends on the difference of boiling points between the components that makes our mixture. And here we have salt and water. Salt have a higher boiling point than water. That's why water will evaporate first. So, water will evaporate first and it will form a vapor, which this vapor will rise out of the distillation flask and it will go through an area of the apparatus, which is here and known as the condenser. The condenser is kept cool by the action of water, which flows around the pipe on the outside of the condenser. Therefore, as the vapor enters the condenser, it is cooled down and it condenses. Condenses just means it goes from the gas back to the liquid. So the pure liquid will be collected in this container that is below the condenser, and any component with the initial mixture with a higher boiling point will be left in the distillation flask in this simple distillation method. So this distillation method, which is the simple distillation one, can therefore be used to separate substances that have significantly different boiling points to each other. If a mixture is made up of 2 similar boiling points to each other, then these liquids will evaporate together if we're going to use simple distillation, and they will condense together and both of them will flow back into this flask here, so there will be no separation of the mixture.